Good morning, students. I am your teacher Hemlata, and we are going to understand the chapter 8 of class 11th biology, the cell, the unit of life. And I am sharing the screen with you. We are going to understand about the basics of cell as unit of life. You are looking at the slide that how brick is made of wall. So brick is made of brick is making the wall. Wall is made of brick. One brick acting as a unit of wall or a house, because from the walls, only four walls, we get a room. So house is built by the bricks and the wall is made by the bricks. The brick can be compared with the cell. So as the living organisms are also made of cells, whether it is a fungus, whether it is plants, different types of animals, these all are made of cells. Human are also made of cells. The first seen with the invention of microscope in 1665. Cell was first seen by Robert Hooke, and they are observed as a honeycomb like empty compartments in the cork. And this was a Leeuwenhoek microscope. Leeuwenhoek is the cloth merchant, and he used this type of microscope, and he made the diagrams of the creatures which are present on the page as shown in the side figure. So he was observing different kinds of organisms, and this kind of picture he made of paramecium, of euglena, and of diatoms, and different types of bacteria. He made those pictures and first observed the living microscopic single-celled organisms in the water, and he called that organism animalcules. So these organisms are called animalcules. These were the first Leeuwenhoek microscope which he made in the 1600s, late 1600s. Robert Brown in 1831 discovered the nucleus. So Robert Brown was the one who discovered the nucleus. In the plant cells also it is seen, and in animal cheek cells also it is seen. It's a dark stained central structure. And in detailed structure through electron microscope, we can find the detailed structure of various cell organelles like chloroplast. This is the detailed structure of chloroplast. This is the detailed structure of endoplasmic reticulum, mitochondria. It is made by, the images are made by electron microscope. This is the electron microscope. You can see the tubular structure, which is getting the pictures of the detailed structures of the animal and the plant cells. All the living organisms are made of cells and their product. This was a statement of the cell theory given by the Matthias Seldian and Theodor Schwann. Rudolf Virchow also added one point of the cell theory, that all the cells arises from the pre-existing cells. So from these both two statements, Schledin and Schwann, Rudolf Virchow also contributed to cell theory. Onion peel cells, you can see, you simply make the diagram of this onion peel and make the slide also in the lab. Onion peel cell, and human cheek cell is observed like the next cell which is shown in the figure. A is the cell membrane, B is the nucleus, and C is the cytoplasm. And we draw the images of the animal cells as shown below, and this is the bacterial image which is shown next to it. Capsule, cell wall is there, ribosomes, cytoplasmic membranes, pili, nucleoid region, prokaryotic cell structure is shown here, flagellum. So plant, animal or prokaryotic cell, cytoplasm is the main area of cellular activity. Cytoplasm, and in the plant and animal cell, cytoplasm is the area where all the activities of cells and take place. Largest to smallest cells, almost same cellular machinery. Sperms are the smallest cells and egg are largest. Prokaryotic cells are there, which include bacteria, blue-green algae, mycoplasma, purulimonia-like organism. Bacterial cells may be bacillus, rod-shaped, coccus, spherical, vibrio, comma-shaped, and spirillum. So the four common shapes of the bacteria are given. Round shape, that means coccus. And the rod shape like this is bacillus bacteria. This is the comma shape vibrio, and the spiral bacteria is the spirochete or spirillum. And this is the basic diagram of the bacteria shown at the corner. Bacteria has three layers: glycocalyx, cell wall, plasma membrane. The slime layer is also a loose covering of the bacteria, and the capsule is the tough covering. Glycocalyx can be as a loose sheet in some bacteria, and which is called slime layer, and glycocalyx may be thick and tough capsule layer. Nucleoid is the main DNA of the bacteria, which is present in the center. Extra chromosomal circular materials are also present, called plasmids, which shows antibiotic resistance in the bacteria. Bacterium, bacterial chromosome, plasmids, mesosome, chromatophores, extensions of plasma membranes are there. Mesosome help in the cell wall formation, DNA replication, distribute to daughter cells. Flagellum have three parts: filament, hook, and basal body. So flagella internal is divided into three parts. These are the parts. You can see there are two rings in the uh, inner plasma membrane, outer plasma membrane also have two rings. And then a hook-like structure, a curved structure hook is present. This basal body is made of um, these two rings, outer membrane, inner membrane, which are present in the outer and the inner membrane, and one in peptidoglycan. And filament is there, which is a long thread-like structure, filament. So three parts make the flagellum, filament, hook, and basal body. Villi in the fimbria, the surface structure which do not play a role in motility but help in attachment. Based on the staining property, gram-positive and gram-negative bacteria can be understood. Gram-negative bacteria has pink color because they have taken the secondary stain safranin and get pink green color, and gram-positive bacteria take first stain crystal violet, so they are seen violet in color. Staining with crystal violet, decolorization, and counter stain with safranin. These are the steps. Ribosomes, 15 to 20 nanometers. Two subunits are there, 50S and 30S. Together form the 70S. The ribosome's size is 15 to 20 nanometer, and it is made of two subunits, big small subunit of 30S and big subunit of 50S. They together make 70S, and it help in the protein synthesis. Polysomes of polyribosomes are present on the mRNA. You can see that how the small subunit and the big subunit is closely attached like a fist, and they are making one complete ribosome. So ribosomes are present here in the cells of prokaryote. 70S ribosomes are present. These are the cell about the cell structures of bacteria. Inclusion bodies are there, which are storing the reserve materials, granules, synophysin, glycogen granules. Eukaryotic cells possesses an organized nucleus with nuclear envelope and have a variety of complex locomotory and cytoskeletal structures. Eukaryotic cells can be unicellular, multicellular, and they in the unicellular they can be in the fungus and the protist, and the multicellular they can be plants and in the animals. Inclusion bodies we have studied. Generalized animal cell is shown here, which is having various structures like nuclear envelope, nucleolus, chromatin. This all three make nucleus and rough endoplasmic reticulum. Then there is ribosomes which are present in the cells, peroxisomes in the cells are there, smooth endoplasmic reticulums are there without the ribosomes, Golgi apparatus are there. After that, lysosomes are present and mitochondria are present. Cytoskeleton is present in the cell and plasma membrane is present. Centrioles are present. These are only present in the animal cells. Generally, generalized plant cells is also given with the nuclear envelope, nucleolus, and chromatin. Rough endoplasmic reticulum is there. Smooth endoplasmic reticulum. Ribosomes are present on the rough endoplasmic reticulum. Then Golgi apparatus is shown. Vacuoles are shown. Rhizome is shown. Mitochondria is there, and plasma membrane, cytoskeletal element, cell wall, and chloroplast is shown. So prokaryotic cells are, the fact is given that they are 10 times smaller than the eukaryotic cell in diameter and about 10,000, about 1,000 times smaller than in eukaryotic volume. Cell membrane is composed of lipids that are arranged in bilayers and it is mainly composed of phosphoglycerides. You can see the common general image of the cell membrane. It's a fluid mosaic model also. The blue colored structure, which is making a bilayer, is a lipid molecule with a phosphate head and the fatty acid tail. And the peripheral proteins are present in between. And some channel proteins are also present here, which is completely uh, in between both the layers, and carrier proteins are there. Then some of the glycoproteins are present, which means the protein attached to uh, the sugars attached to the proteins are called glycoproteins, and there must be some of the sugars, if they are attached to the lipids, are called glycolipids. Polar head is always the face toward outside and non-polar tail is toward the inside. In arrangement, the bilayer, this is the phosphate group, which is a polar hydrophilic head. So it will be exposed towards the outside. And the tails will be sealed inside of both the layers. The tail will be inside. So these are the structure of the fluid mosaic model, cell membrane model. Singer-Nicholson gave in 1972 this model. According to this, quasi-fluid nature of the lipids enable lateral movement of the protein in the overall layer. Bilayer protein can move laterally. Peripheral proteins are there, which are just present on one side of the membrane, which are not passing complete both the membrane parts. The type of protein are peripheral integral with cholesterol, glycolipids, and glycoproteins. They are present. Erythrocyte membrane has 52% protein and 40% lipids. This type of membrane structure is seen of the cell membrane. Function, it is semi-permeable and helps in transport of molecule across it. So function of plasma membrane is that it helps in the same, because of being semi-permeable, it help in moving the molecules across the cell. Small molecules can diffuse. Lipid-soluble molecule can dissolve in the lipid bilayer and can get inside. And water-soluble molecules like glucose have to travel through the ions. Transport of molecule from higher to concentration to the lower concentration is called diffusion. It does not utilize ATP. Transfer of molecules from lower to higher concentration is called active transport, and it utilizes any positive K, positive ATP is pumped, and so ATP is required. It is non-living rigid structure. The cell wall is a non-living rigid structure giving the shape to the cells and protect the cells from mechanical damage and infection. It helps cell-to-cell interaction and provide barrier to undesirable molecules. Cell wall of the algae is made of cellulose, galactans, mannans, and minerals like calcium carbonate. It is non-living structures. Middle lamella is made of calcium pactate, which holds neighboring cell together. Plasmodesmata is a connection of the two cytoplasm of the nearby cells. Consists of the network of tiny tubular structures scattered in the cytoplasm is the endoplasmic reticulum. ER is divided into intracellular space into two distinct compartments, luminal inside ER and extraluminal cytoplasmic surface. Rough endoplasmic reticulum ribosomes are attached to the outer surface continuously with the outer membrane of the nucleus. Function is involved in the protein synthesis. Smooth endoplasmic reticulum lacks the ribosome's functional site for synthesis of lipid. Steroidal hormones are synthesized in SCR. Golgi apparatus first observed by Camillo Golgi in 1898, and transport is occurring here. Two phases of the organelles are the convex phase or forming phase and concave transphase or maturing phase consist of cisternae stacked parallel to each other. Functions is performing packaging of material to be delivered either to intracellular targets or the secreted outside the cell. Important site of formation of glycoprotein, glycolipids, and membrane biogenesis. This is the function of Golgi bodies. Protein synthesis by the ribosomes, and they are made on RER modified in the Golgi apparatus and packed and released. Lysosomes are the membrane-bound vesicular structures formed by the process of packaging in the Golgi apparatus, containing hydrolyzing enzyme, lipases, proteases, carbohydrases, which are active in acidic pH. It is called suicidal bag. It is for intracellular digestion. Lysosomes function. Apoptosis also it is included. Vacuoles are the membrane-bound spaces found in the cytoplasm containing water, sap, excretory products, etc. In the plant cell, vacuoles occupies the 90% space. Function is that the plant stonoplast single membrane of vacuole facilitate transport of ions and other substances. Contractile vacuole for excretion in amoeba and food vacuoles are formed for the protist for the digestion of the food. Double membrane bound structure is the mitochondria. Outer membrane, smooth and inner membrane form the number of enfolding called cristae. Cristae increase the surface area. The inner compartment having the matrix. It is the powerhouse. Production of the cellular energy occurs in the site for aerobic respiration, mitochondria. F0, F1 complex is there in the membrane. Matrix possesses single circular DNA molecule, a few RNA molecules, and ribosomes, 70S, and divided by binary fusion. Plastids are leucoplasts for storage, chloroplast for photosynthesis, and chromoplast for color. And protoplastid ethioplast can be formed by the degeneration. Protoplastid can be converted into other different kinds, three kinds. Alluro and amylo and aleo are the three types of leucoplast found in plant cells and in euglenoids. Chloroplast, chromoplast, and leucoplast are the three types of the plasmids, depending on the pigment contained. Chloroplastid give colors to the petals in the fruits. Chloroplasts are double membrane bound organelle present in the plant cells, and they mainly in the mesophyll cells and of the leaves and various shape, one per cell to 20 to 40 in the plant cell. In the stroma, the space inside the chloroplast contain DNA and RNA. Ribosomes in the grana flattened sac thylakoid contain chlorophyll and trap sunlight for photosynthesis. Stroma lamella connect thylakoid of the grana. Chloroplast, colorless plastid are found in storage parenchyma and other colorless tissues. Myeloplast store carbohydrate, helioplast store oil, and erythroplast proteins. Composed of the RNA and proteins without membrane, eukaryotic ribosomes are ATS, Swedberg unit is S. Function is the site of protein synthesis. Microbody is the type of organelle that is found in the cell of the plant and protozoa in animals. It is also involved in the detoxification of peroxides and in photorespiration in the plants. Microbodies contain enzymes that facilitate the breakdown of fats, alcohols, and amino acids. Cytoskeleton network are the structures which provide the support to the plant, made of proteinaceous molecules and called microtubules and microfilaments. Mechanical support, motility, and maintenance of the shape of the cell is their function. Cilia and flagella are the longer and responsible for cell movement. Cilia are smaller structures which work like oars with the help of movement. These are the internal structures of cilia and flagella. They are covered with a plasma membrane core for axoneme 9+2 arrangement of axoneme microtubules, and central tubules are connected by bridges enclosed by a central sheath, are connected by peripheral doublets of radial spokes. Centrosome, centrioles. Centriosome contain two cylindrical structures called centrioles, surrounded by amorphous pericentriolar material. Here 9+0 arrangement centrioles from the base of the cilia or the flagella. The spindle fibers for the cell division in animal cells, they produce spindle apparatus during cell division. This is the work of the centrosome to produce the spindle fiber for cell division from the basal body of the flagella and the cilia. Nucleus, it has double membrane structure in the between the perinuclear space is also present. Contain chromatin, nuclear matrix, and nucleoli. And size and the number of nucleolus are more when the cells are metabolically active. The outer membrane continues with the endoplasmic reticulum with the ribosomes. Nuclear pore used to communicate nucleoplasm and the cytoplasm. No nucleus in erythrocyte, RBC of mammals, and sieve tube elements in the vascular plant. Chromatin is equal to DNA plus non-histone protein. During the cell division, chromatin coiled into the chromosome. Chromosome DNA plus histone plus protein and non-histone proteins. Chromosome, human cell has 23 pair of chromosomes. Centromere primary constriction, the disc is known as kinetochores. The chromosomes on the basis of position are 4 types: metacentric, middle centromere, submetacentric centromere nearer to one end of the chromosome, acrocentric centromere situated close to its ends, telocentric has terminal centromere. Satellite is non-staining secondary constriction of same chromosome which give appearance of small fragment called satellite. So these are the structures which we have discussed in the PowerPoint. I hope you have understood all the